So let's move on to our final topic for Unit 6, biotechnology. Biotechnology, which is a huge field, and there's a lot of jobs in this field, especially in like bioinformatics and things like that. Programming for a lot of new systems that they have. But let's talk about some classic systems that have been in use for a while. First one I want to talk about is something called gel electrophoresis. And you may have seen this on, if you've watched any crime shows before, and they're comparing different DNA samples, and they run something like this, and they're comparing these different bands here to see if they line up correctly, right? See where these different bands line up to see whether somebody's DNA is the same as somebody else's DNA. Well, let's go ahead and take a look at this and see what how does this work basically. And the first thing you got to know about DNA is negatively charged. That backbone with those phosphate groups are very negatively charged, right? Highly negative, uh, very very negative. So we can abuse that to actually look at DNA and watch it migrate through this gel. So what happens is you have a strip of DNA. Let's say you just have a random strip of DNA. Um, and let's say you send a restriction enzyme that cuts at a certain sequence here, right? A restriction enzyme. Restriction enzyme. It's going to go through here and it's going to do what's going to digest this DNA, and it's going to find a specific sequence. I'll just call it like A T C C T A or something, right? There's going to be some sort of sequence in here that this thing goes through and tries to find on this DNA, and wherever it sees that sequence, it cuts it, right? So you'll end up with kind of this DNA, which you have maybe a medium-sized piece over here, a small piece right here, and maybe a larger piece on the end. Well, if you happen to not have that sequence that it's looking for in a certain location on the gene, you're going to get a different result. Or if you have it somewhere else, right? So, for instance, if this DNA right here doesn't have that sequence in these two spots where it cut on this top one, then you'd end up with one long piece of DNA, right? So this would be subject 1 and subject 2. Subject 1, after you've digested it, it's going to have DNA in which he has a really small piece, medium-sized piece, and a decently large piece. Subject 2 is going to have just this large piece of DNA. And this is a very simplistic look at it. But so what we do is we set up this machine in which we take these, we put this buffer solution in here. We put this gel in here that's basically this medium that DNA can move through, but it's kind of difficult for it to move through at times, depending on its size. So you basically electrically charge this, positive one on the side, negative on the other. DNA, remember, is negatively charged. Since it's negatively charged, DNA is going to travel towards this positive electrode, right? Now, what you're going to find is, and this is, just normal physics, right? If I had you in this, I usually do this in class, but obviously this year we're not in class. So if I had you stand at the back of the classroom and I had all the desks out, kind of impeding your progress, and I asked you the whole class to hold hands and make it as a group to the other side, it would take you quite a while because there's so many of you. However, if you were in smaller pieces, quote unquote, it would be faster for you to move through. So the smaller pieces are going to move further. The shorter bands are going to go farther. The larger bands are going to be too big to fit through kind of these little hole pores and stuff inside of this gel, and they're going to take longer, or they're going to be slower, right? So you're going to find the bigger bands are going to be down this on this end, right? Large bands, and smaller bands are going to be this way. And that's a way that we can kind of look at DNA and compare them to each other. The next natural question you might be asking is, how do we get all of this DNA? How do we get such large quantities of DNA? And we do something called PCR, or polymerase chain reaction. So if you take DNA that needs to be replicated and you want to amplify it artificially, right? Now, our body obviously has normal mechanisms. Cells have normal mechanisms to do this. But if you want to do this in a lab, you want to make billions of copies of this DNA very quickly. Well, we can do that. We can do that. And all it requires is a little bit of heat and a cooling process and a very special uh, uh, DNA polymerase, which I'll talk about a little bit, right? So the first step in this PCR process is called denaturation of DNA, i.e., you're taking DNA and you're heating it up to a very hot temperature, and you're splitting this DNA apart. These hydrogen bonds are breaking down, right? Now, the next process we need to do is annealing. Annealing is the process of primers being added because we have spare DNA primers sitting around in solution. Also, we have nucleotides sitting around ready to be added. So these primers are going to anneal to a certain spot, and there's going to be something called a Taq polymerase. This is a DNA polymerase 3. And it is a very special DNA polymerase, which can operate at high temperatures because remember, we've heated this up a lot. The enzymes in our body, were we to do this, would denature as well, and they would not be able to add nucleotides to a growing strand. This Taq polymerase operates at high temperatures. It was isolated from a bacteria in hot springs. So it can operate at these high temperatures. This Taq polymerase comes along, adds bases, and then you repeat this process again. DNA splits. Primers are added, bases are added, and you can do this to get billions of copies of DNA, right? Billions of copies of DNA. And this is the basic process of how PCR works and how you get all that DNA that you use and what we talked about previously, but that gel electrophoresis. And these two processes were something that I'd like to do in class. If this is the first year I made these videos, 2020, then you'll know we didn't have class and I didn't get a chance to do this, but I did want to do this with you guys in class. Okay, and finally, what I want to talk about again is that bacterial transformation, which you remember from last video. So we have these things called plasmid maps. Right? And base pairs. This is what the BP stands for. This is how many base pairs the specific plasmid is. I went and found just a general looking map here. And you'll see it's labeled in certain locations, right? EcoRI, HIME3, BAM-Hi. These are different spots where a restriction enzyme can come. If you remember those restriction enzymes, this is specific spots where they can come in and act as kind of a molecular scissors here and cut the DNA in certain places. Now, what's nice about this is we can take these plasmids and we can cut them here and insert stuff inside. We can insert stuff inside there. Now, what's nice about that is we can take these plasmids in a lab, we can alter them, and we can allow bacteria to uptake them through this bacterial transformation process, which can be kind of useful for us. And I'll give you one example here. You can take a human cell and you can snip out this insulin gene. You can take a plasmid and you can cut it in a certain spot and insert that insulin gene into that plasmid. Now you have to wait for that bacteria to you have to go through a process to have this bacteria uptake this plasmid. You can grow these bacteria and boom, now you have a process for making insulin with bacteria, right? This is how most of our insulin is made, in large vats, right? With these bacteria growing, and they are essentially insulin machines. They have this gene for making human insulin, and they just make it, and they make a ton of it, and that's essentially how insulin's made through this process of bacterial transformation. So there's some other processes, some more recent processes. Some of you have probably heard about this before. This is kind of it's not really new anymore, but this CRISPR-Cas9 process is cheaper, more efficient way to kind of cut DNA that scientists use a lot now. And this is the main advantage, in that you can run a bunch of experiments that you couldn't before, because this is a very inexpensive process. But I'll have a podcast I'd like you to listen to up on Schoology about this, and I think it's something fun to go through and research, because it is kind of one of the new things that scientists are using pretty exclusively for gene editing and things of that nature. And of course, when I talk about biotechnology, this is barely scratching the surface. I mean, I just went over a few things in a few minutes, but there are many things. We can actually print DNA now, and print exact bases that we want. You can go on a website and say, hey, I want a DNA, a double-stranded DNA that is, AT. So you can come up with the exact code you want, and you can make DNA with that code online and actually order it. So technology has come quite a ways. So don't think this is like the end of it. This is just barely not even scratching the surface. But I think I'm going to leave it at here for this video. I hope that made sense, and thank you for listening.